since we are recording, do some announcements and reminders. The first announcement is that I basically took the weekend off. Which I don't usually do. I didn't hardly get any work done, so if you sent me an email or you submitted something for grading and you wonder why you hadn't heard back from me, that's why. I just basically, like I said, took the weekend off. So I'll get caught up. It's also a good time to remind you too that if you haven't submitted something that you should have, now's the time to do it, because as long as it's submitted before I get around to grading it, I consider that on time in this class, right? So if you haven't done the pre-lab for this week, you might go ahead and do that. What else? Announcements and reminders. I guess that's mainly it. So remind you again, lab is on. It's in person this week on Wednesday at noon as usual. Bring a computer, and if you want, you can go ahead and do it before you come in. I think that's it for announcements and reminders. Anybody have any questions? All right. Actually, I remember somebody asked a question.
I actually had a question real quick.
Yeah.
For the aluminum cans, can you explain that a little more?
Yes. So, and if anybody needs to, you can refer back to the announcements. I'll make sure to post this. I think it's in the announcements, but so yeah, you can get extra credits, like 5 points a week by bringing in at least a Kroger or what's the word I'm looking for? Grocery bag full of crushed aluminum cans. So it has to be full, has to be at least the size of a Kroger bag or a grocery bag, and it has to be crushed. So, that's free points if you guys want to do that. Yeah. But again, the details, everything I just said is written down. So if you forget what I said, you can just look at those announcements. The ones I posted, it says new information, must read, and then under that it says old information, you don't have to read. So you can refer back to that under the old information. So good question. Yeah, you can get free points just for bringing in cans. Also, make sure it's not leaky. So if you need to do more than one bag, double bag it, triple bag it. Please.
And the garbage, do you want us to go pick up litter, or how's that work?
Yeah, that's a separate one. Yeah, so you could also get 5 points a week for picking up trash. I'm just trying to get you guys involved in the community. And the details, I don't even remember. The details are written on that announcement too, because it's sort of like the other extra credit, where you can get extra credit by doing it. And take a picture of you holding the trash bag, and I can tell where you've done. I can tell where you've cleaned up. And then even more points if you're wearing the WVSU, gear while you're doing it. And even more points if you let me post it to social media to help brag on you guys and help brag on the university. So yeah, 2 different things. But yes, so that's the details with that. Also, they're written in the announcements. And obviously, if you ever forget, you want to ask me again, just do. Anything else? All right, let's do attendance very quickly. Let's see, because we're actually running. It's now officially 8. Today's date, 24th. All right, so the first word for attendance will be skeleton. Since there's one in our room now, and since that's what we'll be taking our picture with on Wednesday and Friday. All right, let's see here.
I need to plug my laptop in. I use this.
Yep.
Okay.
And all right, so they're now accounted for. Obviously, if you're online, you're just going to send me the attendance word sometimes before 9 a.m. And that'll be that. So let's jump into it. Back to chapter 10. We finished the first main bullet point, which is great. Now we need to get to this second main bullet point, which is how we go from DNA to RNA to protein. This is probably one of the more important things you're going to learn, to understand so many different diseases, especially viruses, and to understand, vaccines, especially the new COVID vaccine. You need to understand what I'm about to teach you. And notice too, it says from DNA to RNA to protein. It doesn't say from RNA to DNA, right? And we'll get to that. But when you talk about the COVID vaccine, It's an RNA vaccine, and you're going to learn a little bit more about what that means. But RNA doesn't turn into DNA. So when people call that thing gene therapy and say it's going to mess with your DNA, that's not true. And you're going to learn how in this portion of the chapter. But let's jump into it. As you already probably know, DNA provides instructions, right? It's instructions on how to build a polypeptide, right? How to build a protein. And it provides these instructions to cells, right? So sometimes your cells might need a particular protein, and also to the organism as a whole. So sometimes you might need a particular protein. As you already know from the last chapter, excuse me, your genotype is the inherited information, right? You know that. And that is the sequence of nucleotide bases in DNA. So that's the new part for this chapter. You need to know that. So all the information that the DNA is providing to the cell and providing to the organism. How's it doing that? It's doing that in the sequence of the nucleotides. So the same way the English language has words, letters that make up words. Your DNA has letters, right, that make up the words of proteins. So we're going to talk more about that as we move forward. But other questions so far? This is still just an introduction to the good stuff. All right. Let's talk about how DNA provides these instructions. Again, as you already know, a phenotype, those are physical traits from the actions of a wide variety of proteins. So when we talked about phenotypes in the previous chapters, we kind of said it in a different way because we were looking at it in a different context. Back then, we said the phenotype is the result of your genotype. And that is true. But now we're getting a little bit more specific and saying it's from the actions of a wide variety of proteins. So your eye color, for example, right? There's proteins that dictate that, which is why there's genes that dictate your eye color. But your book gives a couple examples too. There's structural proteins that help make up the organism itself, right? So there are actually, you need protein, as you know. There are molecules of protein in your body, but also enzymes, right? And in a sense, that's almost more important because it's those enzymes that are making or dictating the phenotype, if you will, right? And I found a non-book example, I should put on the screen, but I left a note to myself. So the non-book example is melanin. The stuff that makes your skin dark. The TYR gene provides instructions for making an enzyme called tyrosinase, if I'm remembering that correctly. This enzyme is located in the melanocytes, which are specialized cells that produce pigment, right? So it's not the protein itself, right? The genes that dictate your skin color. It's not necessarily that you're seeing protein, but there's enzymes that are making that protein, or the enzymes that are making that the pigment. Anyway, you don't need to know that for the exam. These are just some examples. So any questions? All right, here we get to some important stuff. So if you're taking notes, now it's time to take notes. DNA specifies protein synthesis in 2 stages. You need to know these 2 stages in order, and you need to know what they are. They are transcription and translation. Make sure you know that. Again, everybody studies their own way, but my recommendation is make sure you have that memorized before you add the details. Just know that when we go from DNA to RNA to protein, that whole thing is done in 2 stages, transcription and translation. Then once you have that memorized, then you need to know which is which. So transcription is when we go from DNA to RNA. And then translation is when we go from RNA to protein. And of course, we can't keep it that simple. Later on, we're actually going to look at this in greater detail. There's more things you need to learn about transcription and translation. And for the exam, you'll need to know, there'll be a little bit of comparing and contrasting. Like, how are these 2 things similar? How are they different? So be prepared for that. And your book also points out that this relationship between genes and proteins is one of information flow. And if you remember way back in chapter 1, we said that was going to be one of those main themes of the semester was information flow. So here it is, it's back. We're talking about information flow again. And again, the function of a DNA gene is to dictate the production of a polypeptide. It's not going to be a test question. It's just something you should know, right? Basically, DNA, right, genes provide instructions on how to build proteins. You should know that. So any questions about what's on the slide? I'll even circle that. Just one more time, or circle it just to really emphasize, you need to know that. Transcription, then translation. DNA to RNA, and then RNA to protein. All right. So let's talk about how we go from nucleotides to amino acids, right? The overview. So again, this is going to be an overview. Then later, you can see down here, we're actually going to get into details. Right now, this is just an overview. The information in your DNA, or any DNA really, is transcribed to RNA, and then that is translated into polypeptides, right? So everything I just said earlier, I'm saying it again. And then those polypeptides fold into proteins. If you remember back in chapter 3 when we talked about proteins, when we first learned about them, I said that there's that relationship between a polypeptide and a protein. A polypeptide is just a long string of amino acids, right? It's not technically a protein until it folds up into its proper shape. But here's a a visualization of what we're saying. There's your DNA, right? There's a chromosome inside the nucleus. We need to transcribe that into RNA. So we go through transcription. Now we have a molecule of RNA. That then leaves the nucleus and translation occurs and we have our protein. So again, memorize this before you start adding the details. Notice too, the transcription happens inside the nucleus and translation happens outside the nucleus. That's something else you would need to know for the exam. And of course, you already know DNA is double-stranded, RNA is single-stranded. Let's have that be our next attendance word. I'll circle it, point at it. That is the next word for attendance right there. If there's no questions, I'll move forward.
The slide isn't showing.
It's not showing?
Just you, just the video view is.
Interesting. I'm glad you guys said something.
It zooms in on you and stuff.
Well, that's boring. Let me fix that. I appreciate you guys saying something. Good. And really, this is one of the first things you needed to see, I guess. Anyway, any questions?
The word is circled there, right? It's that word?
Yep, see it?
Okay, gotcha.
Yeah, a little arrow pointed to it. All right. Sorry about that, everybody. Pull this back up. Keep moving. Anyway, if you download the PowerPoint, this is video you can watch. It's a great little animation. I highly recommend it. It's neat because it actually shows what it looks like. So you can see the cellular machine that I call it, and I'll give you the actual name for it later, that clamps onto it, reads the code, and builds the protein. So like I kept saying, or like I said earlier, in the English language, we have letters that make up words, right? And those words can be used as instructions on how to build stuff. Well, it's kind of the same thing here. We're dealing with a language. And what is the language of nucleic acids? Like I've already said, and like you need to know, for DNA, it is the sequence of nucleotide bases. I mean, same thing for RNA, actually. So it's the sequence of nucleotide bases. So the same way A through Z, right? The same way those alphabet for us is what we use to make words. DNA and RNA use the A's, the T's, the C's, and the G's. And of course, for RNA, it's not a T, it's a U, but we'll come back to that. A typical gene consists of thousands of nucleotides. Be careful with that. It's an easy mistake to make. Somebody looked up on paper. It was a good paper. It was a good question about how many genes are on the typical human chromosome. So just got to be careful what you look up. It's easy to mess it up. Anyway, let me cross that out because that is interesting, but I'm not going to ask it for you. I'm not going to ask you that on the exam. So you don't even know that a typical gene has thousands of nucleotides. But it is a good independent work topic, as at least one of you already knows because you've already looked it up. And you can look it up again if you want to fix it. But yeah, instead of just saying the typical gene, get a little bit more specific. Maybe look up some genes. And then how many genes, if we're talking about how many nucleotides are in a gene, then how many genes are on a chromosome? Or how many genes does a typical human have? Or how many genes does a typical, whatever your favorite animal or plant, how many genes does it have? Anyway, moving forward. Like I said earlier, we're talking about a language, right? So the DNA is transcribed to RNA, which basically means the bases of the RNA are complementary to the DNA, meaning this. And this is something you need to know. You're going to see it writing later. But here's just an example. Let's just say AATGC, right? There's a strand of DNA. I'll put a D there for DNA. Now, if we were to transcribe that, when I make the RNA, it has to be complementary to that DNA strand. So does anybody remember what goes with an A? And you really need to know this, so this is good practice. What goes with an A? Okay, that's okay. Tell me now. A's go with T's, right? So there would be a T that, whoops, good grief, and I'm messing up myself. A's go with T's, but keep in mind there are no T's in RNA, right? So wherever there should be a T, you would have a U. If that'll work. Anyway, AU. So again, A's go with T's, and then C's go with G's, and you need to know that. C's go with G's. So that's it. And you will get a question on the exam where it says transcribe this strand of DNA. And I can go ahead and tell you right now, to save you some time, if the answer has a T in it, like the mistake I just made, then it's the wrong answer, right? Because if I'm asking you to transcribe something, that means I'm asking you to make a strand of RNA. RNA does not have a T, it has U. So when you get that multiple choice question, you can just go ahead and eliminate anything with a T in it.
Wait, so are both A's meant to be used?
Yeah. I know it didn't show up there, and that one looks wonky. But yes, as always, A's go with T's or U's, and C's always go with G. All right, any questions? Okay, so that's transcription. Again, just an overview. That's transcription. Translation, which is the next step, is the conversion of the nucleic acid language to the polypeptide language. So hopefully that will be easy for you to remember now, because I'm sure you've all heard of the word translation before, but probably had nothing to do with biology. So when you're thinking about the word translation and you're not talking about biology, you're probably thinking about translating from one language to another, right? Maybe you speak Spanish and you're translating for somebody, right? You're translating Spanish to English, or vice versa, right? You're going from one language to the other. And that's also what's happening here. Because remember, when we go from DNA to RNA, we're still dealing with nucleic acids. We're still dealing with nucleotides, right? That's the same language. But when we go from RNA to protein, we're no longer dealing with nucleic acids. Instead, we are dealing with 20 amino acids. And you need to know that number. But yeah, so that's where we're going from, right? We're going from nucleic acids to amino acids. And it's that sequence of the RNA basis that dictates the sequence of amino acids. So again, sort of like a language. All those A's and U's and C's and G's that are in the RNA basically say, okay, I want that amino acid. Thank you. Now, next you need that amino acid, and it puts it together. So all right, next I want this amino acid, and that's how it works. The letters in the RNA basically tell the cell which amino acid goes next. And it's just a long string of amino acids is all we're making, a long string of amino acids. You already know from the previous chapter that there are 20 of them, and you need to know that for this exam, 20 amino acids. That's a decent independent work topic if you want to look into it. For humans, for example, we need obviously all 20 of those amino acids. Every living thing needs those amino acids, but we cannot make all of those amino acids. So you can look that up. Which ones can we not make? Where do we get them? Anyway, any questions about this slide? The next one is basically a picture of the words that are on the slide. Here's a long strand of DNA, right? There's a DNA molecule right here. It's got a few genes, gene 1, gene 2, gene 3. We're zooming into gene 3 to some part of it, this small section. And we can see the letters, right? The A's and the C's, the G's, right? And then we translate that. Excuse me, good grief. We transcribe that. So like I said earlier, that horrible drawing I made, wherever you have an A, the complementary RNA would be a U. Wherever you have a C, the complementary RNA would be a G. Wherever you have a G, the complementary RNA would be a C. And again, you're going to need to do this. You're going to have something like that on the exam where I say, here's some DNA, translate it, transcribe it, and then here's some RNA, translate it. But yeah, anyway, I'm going to tell you what a codon is later, but again, for now, you can just see that this RNA, right, these letters say, all right, bring me a square amino acid, bring me a circle amino acid, bring me a triangle amino acid. Of course, it's not really like that, but it's just a simplification. Any questions so far? Okay. So the flow of information that goes from gene to protein is based on something called a triplet code, which you kind of saw earlier, and I told you I was going to tell you what it is, so that's what I'm telling you now. The instructions for the amino acid sequence are written in the DNA, right, and then the RNA as a series of words, and we call those words codons, and you need to know that for the exam. You need to know what a codon is. Because again, we're still basically talking about a language. We're still dealing with letters, just like we do in English, except we only have 4 letters to deal with instead of, A through Z. It's only A, T, C, and G. And these words are always 3 letters long, and they're called codons. You need to know that. You need to know that codons are 3 letters long, and that's the words. Those are the words of this language that says, bring me this amino acid, now bring me that amino acid, right? So the codons in the DNA, as we've already said, are transcribed into the complementary codons in the RNA. And then those RNA codons are translated into amino acids that form polypeptides. So it'd almost be as if your chromosomes were like big old books of instructions, right? Books of instructions on how to build stuff. And I'm talking big, like really heavy. And let's say this room is a nucleus. And somebody on campus needs to build something. So they come in because they need that, the set of instructions that you have. You don't want to give them the whole big book because first of all, they don't need the whole big book because they only need one gene, right? They only need to build one thing. Second of all, that's really heavy, so they don't want to take it. Also, it's your only copy, so you don't want to send it out with them. So they come in, and what do? You copy it, right? You write down what the instructions are by hand, right? You transcribe it. And then they take those words that you've written on paper, and then they translate that into building something. Because the words might say, all right, take this 4x4 and hammer it together with this 4x4, right? You're just giving it building instructions on how to build something 3-dimensional. Anyway, any questions about this slide? Here's this slide again. I don't know, this must have been just a mistake. We've already seen this. But yes, again, there's the codons, the 3-letter words that say, bring me this amino acid, bring me that amino acid. So let's talk a little bit more about the genetic code. To me, this is where it gets really interesting, which is, I guess, why I studied this in grad school. Genetic code is a set of rules that converts the RNA sequence to amino acids, right? So everything we've been talking about, we've been talking about the genetic code. Now you know what it's called, the genetic code. Of the 64 triplets, so again, in this language, words are made up of 3 letters, right? And we call those words codons. So really, I don't know why it says triplets. I took that from the book. Another thing to say is, of the 64 codons, that'd be a better word because that's the word we're using. Of the 64 codons in this language, 61 of them actually code for amino acids, and 3 of them say, all right, we're done. Stop the process. We're done. I'm going to put an X through this because you don't need to know those numbers. But for your own knowledge, you now know that some of them actually say, okay, I want this, give me this amino acid. And some of them actually say, stop doing what you're doing. We're done with this process. We're done translating. That might be a good, I don't know, the 64 part, but that might be a good extra credit question. How many words are in this language? 64 words. Of course, if you're a thinker, hopefully this is giving you a little bit of a red flag right now, and you're like, hold on. 64 words, 61 of them code for amino acids, But I told you earlier, there's only 20 amino acids. So hopefully you're thinking, well, how does that work? If these things say, bring me this amino acid, bring me that amino acid, how is it that we have more words than we do amino acids? And that's where this comes in. Make sure you memorize this. I'm just kidding. You don't need to memorize this. But first of all, let me address what I was saying earlier. The reason that there's more words than there are amino acids is because sometimes you might have multiple words that make up an amino acid. For example, GCU says bring me alanine, but also so does GCC, so does GCA, and so does GCG, right? Any version of that, and you're going to get an alanine. So again, there's nothing to study here. You don't need to memorize any of this, but there's a question on the study guide, and there might be a question on the exam where I give you this. And instead of just saying transcribe this or translate this, you might have to do the whole thing. So you transcribe something into RNA, and then you take that code and you tell me what the sequence of amino acids is going to be. So I'm randomly going to write down something. Let's say, I don't know, CUUAGC. It's random. So if I give you that and say translate that, then you're going to use this code right here. Okay, CUU. So CUU. So leucine is the first one. And then AGC. So AGC. And then serine would be the answer. You guys have that? It might look complicated because there's a lot of information on that wheel, but it's not complicated. It's pretty simple. No questions. All right, let's move forward. Here's where it gets even more interesting, I think, anyway. The genetic code is essentially universal, meaning it's shared by everything living on Earth that we know of, whether it's a simple bacteria, something complex like us. It's universal, which basically means that one species can produce protein from another simply by translating the DNA. And that's where we get things like insulin, like I mentioned earlier in this chapter. It's also where we get things like GMOs. Some people are scared about GMOs, but mostly because they don't know anything about them. For example, there is, of course this is a different kind of example. But anyway, there's an apple where they took the gene that makes it turn brown, and they just took the gene out of it, right? And people are afraid of that. There's something, I forget the name of it. It's not super rice, it might be called super rice. But anyway, rice is a staple in some parts of this world. So what they did was they took the genes for beta carotene that are in carrots, and they put that gene into the rice plants, and now those rice plants produce beta carotene, so that's giving them more nutrition. I wish I could do this. I want to take the genes that produce THC and put it into actual food. I think that would be great because people buy gummies and edibles, so why not just make, I don't know, salsa that'll get you high. I'm not condoning it. I'm just saying I want to make it and sell it and get rich. But anyway, you can look that up too. So if you're interested in that kind of thing, and like I said, I think it's really interesting. You can look it up and for independent work. All the different ways we can mix and match genes from different species. And I'll actually give you a few examples on the next slide. Anyway, before I show you that, I'll point this out really quickly. Your book points out that the universality of the genetic code suggests that it arose very early in evolution. So as we're going to learn later when we talk about evolution, the fact that we all have the same system basically is one of the evidence that points towards evolution. Anyway, here's some examples. One of them is not real. You want to guess which example is not real? So here we have some glowing trees. Here we have some glowing pigs. And here we have, I think, a glowing baby rat. Anybody want to guess which one is not real?
The rock.
Actually, it's the trees. You would think, right? Because it doesn't look real. But no, it's the trees. They haven't done that yet. They've talked about it. But yeah, so they've taken genes from jellyfish and other bioluminescent creatures, and they put them in things like pigs and rats. And it's not just to see if they did it. No one's going to get funding. The scientist isn't just going to be like, I would love to see a glowing cat. I don't need money for that. No, but it's actually used for research purposes. But we don't have time to get into that. So yeah, look into it if you want for independent work, all the different ways we can mix and match stuff. So if there's no questions, we can finally finish the overview and get into the details. Now we can actually talk about transcription in detail. Let's jump into that. And the next word for attendance would be pin. Something you write with, like something right here at the bottom of that screen. So as we've already said, and as you need to know for the exam, transcription is the transfer of genetic information from DNA to RNA. You need to know that. You need to know that step one is transcription, and that's going from DNA to RNA. When this happens, the DNA strand separates where the process starts, which is similar, if you remember, to DNA replication, right? Because the code is on the middle. Those things are hydrogen bonded to themselves. So all we need to do is open that up. You need to unzip it, so to speak, so that the code can be read. One of those strands serves as the template, and the other one's not even used. Of course, you don't necessarily need to know that for the exam. And if you don't remember that, you can still understand how this process works. So that's the only reason I'm putting it next to this. In the context of you understanding what I'm about to tell you, you don't necessarily need that bullet point. The RNA nucleotides take their place along the DNA template by forming hydrogen bonds with the DNA nucleotide bases, which is just another way of saying, basically, this is how the same way it worked for our DNA replication, right? We opened it up, the cellular machinery came in, remember we called it DNA polymerase. They came in and read the code, one by one they were added. It's basically the same thing for now. At this point in how I'm describing it, it's basically the same thing. Any questions about what's on this slide? Okay, as you already know, and as you need to know for the exam, the RNA nucleotides follow the same base pairing rules, right? A goes with A and T go together, C and G go together. Of course, if we're dealing with RNA, there is no T, that's a U. And this is all done by something called RNA polymerase, and you need to know that for the exam. So far, for this chapter, I've given you the names of 2 different enzymes, and you need to know each one of them. DNA polymerase makes DNA. RNA polymerase makes RNA. It's pretty simple. This is kind of what it looks like. So you can see this strand of DNA right here. This big old thing that's attached to it, that's the RNA polymerase. And this little thing, squiggly thing coming off of it, that's the new strand of RNA that it's making. So RNA polymerase. I want to point out again, too, that we're giving you the 100 level version of this story and the non-biology based version of the story. It's actually a little bit more complicated. As you can see, that RNA polymerase has different colors. Reason is, not that you need to know this, because again, your book is just telling you, hey, it's RNA polymerase. But just so you know, RNA polymerase is actually a number of different proteins. So it's not as simple as just this one protein. You can look that up for independent work if you want. But for you guys, you can just think of this one protein, this one enzyme called RNA polymerase. The next slide is a better picture of this. Does anybody need this slide still? All right. Here's a picture of how it looked. Well, it's not really how it looks, but it's a good visualization of it working. So you can see the DNA strand has been opened up. The RNA polymerase is attached. It's reading the code. So here's a T. It brings in an A. Right? That A is going to hydrogen bond there. It's going to covalently bond there. Next comes a U, right? Because the U goes with the A. So that comes in. You can see here, there's another A. So we're going to bring another U nucleotide. And it just goes on and on. And this thing literally would just move down the DNA strand, reading it. And as it does that, the RNA strand will just get longer and longer. Any questions about that slide? All right, let's make it a little bit more complicated. More stuff for you to memorize for the exam. Transcription itself is broken down into 3 stages, and you need to know the 3 stages, and you need to know them in their correct order. Initiation, elongation, and termination. Technically, the longer version is initiation of transcription, RNA elongation, and termination of transcription. But I just put it in bold, the stuff that would suffice, right? Initiation, elongation, termination. Hopefully that's easy for you to remember, right? It makes sense. Obviously, initiation is going to be the beginning. Terminator or termination will be the end. And by process of elimination, elongation is the only thing left. Of course, we can't keep it that simple either. So what we're going to do is go into detail about each of these three stages. Because the test question might not just be, all right, what are the three stages in order? It might be, I describe one of the three stages and you tell me what it is based on the description. Speaking of which, here's something else you need to know. The start transcribing signal, right? So where the RNA polymerase knows where to go on the DNA, that is called the promoter. You need to know that. Promoter is where this whole process starts. So remember, initiation is the name, is what we call this process when it starts. But physically where it happens on the DNA, that's called the promoter. It's located at the beginning of the gene, and that is where the RNA polymerase attaches. You need to know that for the exam. And basically, that's all initiation is. Initiation is the attachment of the RNA polymerase to that promoter that I just told you about. And that is the start of the RNA synthesis. So for initiation, it's simply the attachment of the machine that's doing the job, which is the RNA polymerase, right? The RNA polymerase attaches to the promoter. Any questions about that? All right, you guys are pretty easy today. Let's talk about the second phase, which is elongation. Yep. Elongation, as you might imagine, is just basically the RNA is getting longer, right? So you start the thing, that was the last phase we talked about. Then it happens, which is when the RNA grows longer, which is what we're talking about now. It's pretty simple, right? And as this is happening, you can see here that the RNA is kind of peeling away from the DNA. Because you can see here, it's attached to the DNA because that's how it brings it in, right? Nucleotide by nucleotide. But then eventually it peels away from the DNA. And of course, you can see here that the DNA does come back together. It's not like it just opens up and stays open. No, it comes back together. And I'm probably not even going to ask that much detail in the exam. Basically, you need to know the second phase of transcription is called elongation. And what happens in elongation? Oh, the RNA is elongating, right? The RNA is growing longer. Any questions about that? Okay, the next slide, which will also have the next word for attendance. Next word for attendance will be terminator, like that word right here, in this picture right here. The third phase of transcription, the last phase of transcription, is called termination. As you already know, it's very simple, it's in the name. And what happens in termination? Well, there's a DNA sequence called the terminator. That signals the end of the gene. So in a sense, you could say the terminator is the opposite of the start codon, excuse me, the promoter. That's a mistake that's easy to make. We'll cover that later. But as simple as this may seem, it gets a little bit more complicated when I add RNA to it. Because then you're going to have to differentiate between this and translation. You'll see what I mean when we get there. But yeah. So at the end, there's a signal, right? There's a part of the DNA that says, all right, job's done. So at that point, the RNA polymerase detaches itself from the DNA. The RNA goes on and does its thing, which we're going to talk about later, and the DNA strands rejoin. So pretty simple stuff so far. So any questions about termination? All right, here's a picture of everything in one as far as transcription is concerned. Yeah. So yeah, the next picture I'm going to show you is going to show the RNA polymerase attaching to the promoter. It's going to show the RNA polymerase moving down the strand of DNA and the RNA growing longer until finally the RNA polymerase hits the terminator and then it detaches. Here it is. So again, just like I said, RNA polymerase attaches to the DNA right there at the promoter. That's what we call initiation, right? Then it starts moving that way and reading the code. It's opened up the DNA, right? And it's making RNA. And and that is called elongation. Finally, the RNA polymerase, you can see this in red, it hits the terminator, so it detaches, the DNA strand goes back together, the RNA goes on and does its own thing. And of course, like I said, that's what we're going to talk about. As far as we're concerned, it's all about the RNA, this conversation, because again, we're talking about going from DNA to RNA to protein. Well, we've done the DNA to RNA. Now we have our RNA, so we need to talk about that process of making it, turning it into a protein. Anyway, are there any questions about this slide? Okay. You download the PowerPoint, you can see an animation of everything we just talked about. Here we go. Now we're going to talk about the processing of the RNA. So of course, again, it can't be too simple, right? We have to process this, we have to make it a little bit more complicated, have to make the exam a little bit harder. So before we can go from, because we're done with transcription basically, right? You know that translation's next. So before we can get into translation, we got to talk about something else. Sort of like when we talked about cellular respiration, and I kept saying, we go from glycolysis to citric acid cycle. But of course, there was that little middle spot because we had to get the pyruvate groomed and changed so we could go into the mitochondria. It's sort of the same thing here. We've got to make some changes to this RNA. So your book points out that prokaryotes have no nuclei. As far as we're concerned, when we're talking about prokaryotes, you might as well just think of bacteria, right? So bacteria, very simple, they don't have any organelles, including nuclei. Therefore, for them, the RNA is transcribed from the gene, and then it immediately functions as what we call mRNA, messenger RNA. It does that right away for prokaryotes. Because it's simple, right? Now for us, eukaryotes, it's a little bit more complicated. But yes, before we move forward, make sure you understand what this is, mRNA. That is the RNA that we make, right, when we transcribe the DNA. You need to know that. Know what messenger RNA is. And that sounds easy until I give you all the other RNAs that you need to know. You're going to need to know about something called rRNA and tRNA. And of course, this mRNA. And you're going to need to know the difference between the three of them. So anyway, yeah. For the slide, for the exam, the most important thing on this slide is you need to know what messenger RNA is. And then, moving forward in these next slides, I'm going to tell you how we change the mRNA, how we do things to it, because we are eukaryotes. So are there any questions so far? All right. You do not need to know what 3' or 5' is. Don't worry about those numbers. But you do need to know this. One of the ways that we groom the RNA, if you want to call it that, one of the ways we change it, one of the ways we modify it, whatever word you want to use, is something called cap and tail. So you need to know that. Before the messenger RNA can leave the nucleus, it needs something called a cap and tail. What is the cap and tail? It is the addition of extra nucleotides to the ends of the RNA. So you can see here, they have got the cap in red, they've got the tail in blue. Those are extra, right? So we had the RNA that we directly transcribed from the DNA. That was one thing. Then something else came in and I put extra nucleotides at the end. Why? And you should know this. It is to protect it from attack by enzymes and help the ribosomes recognize the RNA as mRNA. We'll talk about that later. But for now, you should just know the cap and tail is there for protection, and also because the machine that's going to do the translating, it needs that cap and tail. So, oh, okay, this is what I'm working with. So, any questions about this slide? All right, let's talk about the other kind of processing that the RNA goes through. And that is RNA splicing. What does RNA splicing does? It removes the non-coding nucleotides from those that do code for amino acids. And I didn't tell you that, so this is new information here on this bullet point. What I did tell you was not all of the codes, not all the words, not all the letters are used. Some of it is what some people call junk DNA. We're learning it's not going to be junk. That's a whole different discussion too. But you need to know this. You need to know the difference between an intron and an exon. because it is the introns that we get rid of. And for our purposes, I guess you could think of it as junk DNA. Again, for our purposes, it might as well be junk. By our, I mean us talking about Bio 101. So that can be confusing. Exon, intron, which one's the one that we keep, which one's the one we get rid of. But here's how you can remember it. The exons are the parts that are expressed, right? We express the exons. That's why we keep them. The introns are the parts in between. That's why we get rid of them. So make sure you know the difference between exons and introns. If you want a little extra credit, or in person, send me an email and tell me the difference between intron and exon. Doesn't have to be, complicated. It could be literally as simple as I just said. Exons are the part that are expressed, introns are the part that are in between, so we get rid of them. All right, any questions about the exons and the introns and the RNA splicing? All right, it gets a little bit more complicated yet again. So that's pretty simple, right? We just got rid of all the introns. However, hold on. Here. Yeah, let me back up a step before I even tell you that. I like that your book points this out. RNA splicing plays a significant role in humans, and that allows us, with our about 21,000 genes, well, I gave that answer away. I guess you could still look it up if you wanted to, and just maybe you could get a different answer or a more specific answer. But anyway, we have about 21,000 genes, but we produce many, many more proteins, which should be interesting to you, I think. You should be like, wait a minute, how does that work? Because the genes are instructions on how to build proteins, and that's the only way we build the proteins is with those genes. How is it that we make more genes or make more proteins than we have genes? And the answer is alternative splicing. Do I have that on here? Excuse me. Yeah, alternative splicing. I'll come back to that. I'm not going to ask you any questions about it, but it's, in my opinion, it's pretty fascinating stuff. It's just worth keeping in the back of your mind. So again, here's the exons, right? That's the expressed part. That's the stuff that we're keeping. Here's the introns in between them, right? And what I showed you earlier was the simple thing, right? We just get rid of all the introns. That would be this, right? So then exon 1 is attached to exon 2, which is attached to exon 3, which is attached to exon 4, which is attached to exon 5, right? Nice and simple. However, there is alternative splicing. So you can see here, this one, when we spliced it, we also cut out exon 3. So from here to here was completely cut out, right? And then on this one, we left out exon 4. So from here to here was cut out. So it's still one gene, but we're putting it together in different ways. And because of that, we have 3 different proteins. Hopefully you guys can see that, right? Protein A, protein B, protein C. Of course, those pictures probably don't make any sense to you because we haven't been taught that, but you can tell they're different, right? They look different. They have a different shape. And as we know about proteins, it's all about the shape, right? So different shape, different function, different proteins. So one gene, multiple proteins from it because of alternative splicing. Anyway, are there any questions about alternative splicing? I'll back up then one slide and show you this picture that I skipped over. So this is just the regular splicing. Again, your book doesn't talk too much about the alternative splicing. It says it exists, but your book just focuses on the easy stuff, right? We keep the exons, we get rid of the introns, we bring it together, and then it's ready to go. Also, again, to remind you, we talked about cap and a tail. So now we have the mRNA. It is now ready to leave the nucleus and go out and be translated. So are there any questions about that picture? All right, when we get back on Wednesday, well, let me do one final word for attendance. Let's just say agent. The bottom word there with the slide. That'll be the last word for attendance. But yeah, when we get back, then we'll get into translation. You guys have a great week.